I met with Kenichi Matsubara, and their genome project was going to sequence full-length cDNAs. And those of you who have done early cloning, you get a radioactive probe for the gene you're interested in, and you try and find it out of the millions on the plate. And I thought, you know, it has to be there someplace. I had the very first automated DNA sequencer from Applied Biosystems. I sequenced 2 receptor genes with it. And I thought, one day I just got the idea, why not just randomly select clones off the plate and sequence them? And it was stunning. I think the very first paper we discovered 2,000 new human genes.
But by doing that, by applying technology in this new way to do science faster, I mean, you started to create an uproar, not the first uproar, not the last uproar, I think. Not the last uproar, because it changes how science is done, what it's used for, and kind of who gets to do it. So, what was the early feedback about your use of these methods to just make gene discovery go quicker?
Well, it's the first time Jim Watson started calling me names. He was very afraid the cDNA approach was going to steal the budget for the genome project. Even though in the end, the genome could not have been annotated without ESTs. That's the main way genes were discovered. Very few people acknowledge that, but that was the data that was used by everybody to annotate the genome. But he was upset with it. Several professors from Harvard wrote a letter to the NIH director trying to get my research stopped because it was unfair. I was discovering genes in one day that would wipe out several PhD theses. And so progress in science and moving things forward isn't always greeted with open arms. But it started the whole approach that led to shotgun sequencing, and it's what we're talking about von Neumann and others. Randomness is a key aspect of that. We're just randomly selecting clones. For shotgun sequencing, it was randomly selecting clones, only all the clones were from a shattered genome. So the aspect of randomness is a key that led to all these other things, including the ocean sequence.
Right. Let's pause on the human genome. In 2000, you were invited to the White House for an event with Bill Clinton. This was the calling of a truce or a tie between the public project and Craig's company, Celera, which had raised $1.5 billion and bought a lot of sequencers and pursued, of course, the shotgun method, just shatter the genome and sequence the bits. So in the book, very early on, you say it clearly, you say you won that race. So tell me why you think you won it and why it is that you agreed to a tie.
So the actual White House event was based on when the Celera computer finished assembling the first genome. It was not based on anything from the public event. It was an arranged temporary marriage. Ari Petrinos is the one who was instructed by the White House to try and get Francis and I to talk to each other, and he invited us to his house. We had all these secret meetings over cocktails. I was a little skeptical about it. But Celera clearly won the science race, but we felt the politics was equally important. A lot of my colleagues, including most of my employees, wanted us just to announce that we won and embarrass the public effort, who was, as you know, you published a lot of the quotes. They weren't exactly friendly, scientific, collegial quotes. It was called, Watson called me Hitler. We were told it was the
Mad Magazine genome.
The Mad Magazine genome. All kinds of things that it moved quickly out of the scientific discourse realm, which is because they were just frustrated because they couldn't compete. They were working on a 10-year public distribution project, like building a highway, we sequenced it in 9 months. But every genome since then has been done with our method, basically. And because the mathematics were so powerful, we'll be talking to Gene Myers later, you know, he and his team are the ones responsible actually for having the genome, because they developed the new algorithm to put 25 million fragments together, which everybody thought was impossible. Gene was one of the ones certain early on because he wrote some test algorithms, you know, that's why we had the gene. The late John Sulston wrote in his book at the White House event, they were a bunch of phonies because they didn't have a genome yet.
Let's get to the book, The Voyage of the Sorcerer 2. So by 2003, the genome has been sequenced, at least the drafts, and you are finding yourself in the Sargasso Sea in the Caribbean on a sailboat and ready to sort of start sucking in gallons of seawater for what is, I think, you know, the first large-scale microbiome project. So how did you end up there on the sailboat, ready to sequence the ocean?
So the notion after Haemophilus was that the algorithms were very powerful. After sequencing Drosophila and the human genome, we knew that they were, but also early on we got everybody sent us genomes after the science paper in '95. And we were sent this clinical isolate from Norway, from a patient refractory to antibiotics. And we sequenced the sample, and the algorithm assembled 2 separate genomes. So it turned out to be 2 related species instead of just one. And one was antibiotic resistant and the other wasn't. And that really proved to me how truly powerful the mathematics were. And that, you know, after the human genome, where we dealt with 25 million fragments, I assumed that we could do very complex mixtures. But there was only one other person on the planet that believed me, and that was Ari Petrinos, head of DOE biology at the time. We submitted a grant to try this in the Sargasso Sea. Every reviewer turned it down with extreme prejudice, but Ari overrode them and funded us to do the experiment. And we just filtered a barrel of seawater, sequenced everything off the filters, and discovered roughly 2,000 new microbial species, and we stopped sequencing after 1.5 million new genes. This was in an environment that people called an ocean desert. Up till genomic sequencing, microbiology the tools were a microscope and what would grow in culture. If it didn't grow in culture, it was deemed not to exist. They're up to the 7th microbe discovered in the Sargasso Sea. It was SAR-11. And it just seemed absurd, and that's why we thought we tested in a desert, just in case the mathematics couldn't handle super complexity. But it turned out it had the same complexity in the desert that it did everywhere else in the ocean. So that was the start. Once that experiment was done, it's just asking simple questions. Was the ocean a homogeneous soup? Or would we find different components in different places? So we followed the Challenger expedition, which in 1870s took samples from the bottom of the sea around the world. The brilliant people in science then said there was no life under 18,000 feet. It couldn't exist. And they found life everywhere.
They were looking for macroscopic life.
And we had the technology. We had new tools. The tool was genome sequencing. So when we were in the Galapagos, everywhere, you know, we were visiting the same places that Darwin visited, but with a different tool set that, you know, he couldn't even imagine at the time. And we saw a totally different universe. But every 200 miles, 80% of the sequences were unique. And so the diversity just kept going up. Physicists started to join us. because there's more microbes in the ocean than stars and galaxies in the universe. So it's just a huge, huge number.
Right. The numbers are in the book. I think, liter of seawater, maybe 1 billion bacteria, and even more viruses. So, I mean, this is shotgun sequencing of the ocean, of everything that is alive. So, I mean, what did you find? What can be learned from these data?
So, one of the most interesting findings, we developed a tool at the Institute where we had a reference genome. For example, with SAR11, we sequenced that genome because it was isolated in the culture. In this, like what happens with analyzing the human genome, like my reference genome, people use Illumina, other sequencing techniques to get fragments that they compare back to the genome. They don't sequence them independently, which we should talk about. But with this fragment alignment, we found that instead of the ocean being comprised of lots of individual species, there were clouds of maybe 10,000 or more related genomes. So the gene order was essentially the same, but up to 50% sequence diversity. We only have about 10% across all mammals. So we had this tremendous sequence diversity with very similar organisms, all photosynthetic. What we found is microbes exist in these huge clouds of related organisms. Retrospectively, the two biggest mutagens are UV light and oxygen. And so these organisms were exposed to that. They're constantly mutated. One of the other major discoveries was that viruses didn't want to kill off their host. They regulated their populations. But the most interesting finding was that the viruses actually carried new versions of photosynthetic genes and would update the software in these cells that provide 50% of the oxygen that we breathe. So dealing with science philosophy, it kind of makes you wonder who's in charge. So the viruses are keeping all these organisms alive because that's how they reproduce. But the viruses are not alive themselves. So it's a strange phenomenon, and maybe viruses are keeping us alive so they can keep reproducing as well, and we're just larger hosts.
So you think viruses arguably are at the top of the food chain?
It's, you could argue that intellectually.
Yeah. So your co-author, David Ewing Duncan, has written an article in Scientific American, and he links the voyage to concerns about climate change, about global warming. So, I mean, as you sailed around the world, this is actually a journey that gets carried out in a series of steps over a number of years, but it's a partial circumnavigation of the world. Like, what was your impression of the planet? How are we doing, and what does the microbiome of the sea tell us?
So it went from just sheer beauty and wonderful crystal clear water to sailing into islands that were miles wide of plastic and trash. And now we have these anoxic zones, the largest ones the size of Africa. Fish and mammals can't survive there. There's no oxygen. Pollutants, all the trash, toxins from toxic algae have killed off all the oxygen producing organisms, and these are expanding. You know, we're all concerned about sea level rise, and that is important, but if we cut off our oxygen production, we have a much bigger problem.
Is it something like 40% of the oxygen is made
40 to 50% by these organisms, and the rest by ocean algae that moved into a eukaryotic cell and became plants. So it all has similar origins, similar biochemistry, but we can't afford to lose 50%. And 1 degree temperature change has a big impact. Those of you who've been to the Seychelles, they have beautiful white sandy beaches. Those are a result of coral bleaching from a 1 degree centigrade change that wiped out a couple of microbes that were symbionts with the coral. The coral couldn't survive without them, and it wiped out one of the largest coral reefs on the planet. So in Maine, we see temperatures increasing. Canadians like it because the lobsters are migrating further north, but could wipe out a multi-billion-dollar industry in New England. So 1 degree is a big deal in the ocean, and we're potentially facing worse than that. So we don't actually know what the outcome is going to be, but we sort of have a baseline from all these samples around the world, and there's been dozens and dozens of smaller and different expeditions collecting more and more samples and data, and it's clear that it's changing. These microbes also drive the weather. If you look at the satellite maps during El Nino or El Nina, the amount of chlorophyll in algae totally changes, and changes that's a driving force for the weather, not as a result of it.
People should read the book. It has a lot of good stories. Maybe you can share one. I think Craig was accused of being, some kind of sea pirate, biopirate, genome pirate while he was on his travels, and there was a couple of close calls. I mean, what story can you tell us about the challenges of collecting these specimens?
So we had to have permits from every government. We had a team that worked with the State Department to get these permits, working with their government and quite often their military. But we ran into geopolitics sort of unrelated to the sampling itself. In French Polynesia, there was a battle between the Tahitian government and Paris over who really controlled the waters there. And the French threatened to sink us, which we took seriously because they sunk the Rainbow Warrior, the Greenpeace boat, protesting their nuclear testing. It turned out secretly they were worried that we would do microbial testing near their nuclear test sites and show the extent of genetic damage that was caused. We had no intention of going there because there was actually too much radiation still. The other place we ran into geopolitics was in the Indian Ocean. The British arrested us in Chagos Island, which we were just visiting. We told the State Department we weren't sampling, but they were afraid because of my notoriety, we'd bring attention to that island chain where the British and Americans removed all the natives that have been suing in world court and have won multiple times the right to go back. People in the U.S. know it. Diego Garcia is one of the islands. We have a B-52 base there, and the U.S. is not about to give that up, so the natives can't get back there. They're worried me being there is going to bring attention to it, so they had to bring an idea to arrest me, and that wouldn't bring any attention. But we had to negotiate through the U.S. ambassador to Britain with the British government to get our passports back. We were held at gunpoint, and I had to sign an agreement saying I broke a law from the 1800s that we had unlicensed dive equipment aboard a sailing vessel. That's the best they could come up with for arresting us, but they had guns and we didn't.
Yeah, close call. Craig, I want to bring it up to the present. You have a company called the Human Longevity Inc., which I think is involved in sort of one of the great projects of genomics now, which is to link genes to phenotypes. So tell me what the company is and what you're trying to do there.
So it's a clinic set up to do the most comprehensive, non-invasive phenotype testing. The only invasiveness is a blood draw. It came about because all the people analyzing my genome noticed that I had a variant in ApoE that increased risk for Alzheimer's disease. So I asked my friends in neurology at UCSD to get an MRI brain scan and MRI PET for amyloid. And they couldn't do it because I didn't have any symptomology. And this sort of led to my notion about doing pre-symptomatic testing. They finally agreed to do it under a pseudonym. I suggested Charles Darwin, and they said, no, that's too obvious.
That's taken.
That's taken. So, they made up some name, but I had no amyloid in my brain, and they said I had a brain of a 40-year-old. And that's where it first became clear that predictions from the genome don't apply to individuals, even though they should. And a lot of the genome literature is based on looking at patients with diseases, not screening the broader population. And so we set up to do large-scale phenotype testing, whole body MRI, brain scans. MRI gives you a test that you can't get too many other ways of your organ fat, which is a very important diagnostic test. And the long and short of it, we only tested people that thought they were healthy. We found out 50% of them were not. 5% over 50 had a major tumor that they were unaware of, including myself and my colleague Ham Smith. Both of us are alive because we went through early MRI testing. 1% of everybody in the population in this room has a brain aneurysm. And is skewed towards people under 50 because of genetic selection. People die from brain aneurysms, so there's very few older people with brain aneurysms. They can be treated in 15 minutes as an outpatient versus just waiting to die from it. So pre-symptomatic testing leads to cures for all these tumors and diseases, but the medical establishment is now anti-Greig because they get paid for solving people's symptoms, and a lot of them think it's wrong to do testing on people that don't have symptoms.
Right. Which genome are you talking about? Are you talking about the nuclear genome or the cancer genome that's in the blood? I mean, are we talking about a liquid biopsy? Because in population genetics, it's the law of big numbers, right? Many genomes or many genotypes and many measurements of phenotype. You have a more in-depth phenotype but fewer numbers. So how do you escape the law of big numbers? Or can you?
Well, the big numbers actually help. So we published a paper analyzing 10,000 genomes once we had them, but we also sequenced the cancer genomes and tried to make specific cancer vaccines to the neoantigens, the mutated genes that showed up in cancer. And the interesting phenomenon was that these were different in virtually every cancer. But one of the most important things, the late Tom Caskey, who was one of the best human geneticists on the planet, individually reviewed every data on every patient. And we were able to make correlations quite often between the phenotype, the disease that we discovered in somebody, and some change that was new in the genome that people were unaware of. So if the genome field switched to doing phenotype testing on every genome, genomics would move ahead multiple orders of magnitude faster. Sequencing more genomes is great for population tracking. It's not leading to understanding of the genetic code and how you're different from me and whether you're going to have unique diseases versus my life outcomes. We need specific phenotype testing along with the genome, and the genome field still doesn't understand that 20 years later.
Yeah. We're running out of time. Craig, I am different from you. There's nobody like you. And I just want to end, reporters in the room can pay attention to this one. Just want to end with a question. Were you approached by the Trump administration to become NIH director? Is that a true story?
It is a true story. They badly wanted to replace Francis Collins. I was asked if I would do it. And I said, if you'll set aside 10% of the NIH budget for new research ideas, I will consider it. And they said, well, you know that's not going to happen. I said, can you at least let Francis know I was your choice?
Terrific. Craig, thanks for being here. We'll see you around the festival. Everybody have a great 2 days. Thank you so much.
Thank you.